Welcome back. This is where we left off after part one. And this is the table I said has a lot of the new vocab words, some that we've introduced already, like fascicle and skeletal muscle fiber, along with a lot of other new ones that we're going to introduce in this second part. So we're going to really zoom back in and talk about the functional unit of muscle, and that's going to be the sarcomere. So we're going to talk about how our muscle works and actually contracts. All right, so before we really talk about how it works, we have to define some of our new parts here. So we mentioned this earlier that root my slash o, myo, is for muscle, and sarco can also mean muscle. So you'll see here we have a couple of specially named organelles that are specific to our muscle fiber. So if you remember back from unit one, we had a plasma membrane or the cell membrane that surrounded a cell. When it comes to muscle fibers, that is the sarcolemma. So the sarcolemma is the plasma membrane of a muscle fiber, and that sarcolemma is going to surround sarcoplasm. So that's the special name for cytoplasm when it comes to our muscle cells. This is something that we have not introduced yet, but this is an inward fold of the sarcolemma. So this is building on the sarcolemma, kind of a portion of the sarcolemma, and that's the transverse tubules, more commonly referred to as the T tubules. And so this is an inward fold of the sarcolemma, and this is how we project into the interior of the muscle cell. And when we reach the neuromuscular junction section, you'll see why this is important for being able to activate our muscle fibers. In Unit 1, we talked about the endoplasmic reticulum. There was a rough one responsible for proteins and a smooth one responsible for fats. For our muscle cell, we have the sarcoplasmic reticulum. And so this is a specialized version of the smooth ER. And the big thing to remember here is that it stores calcium for our muscles. And we'll get into why our muscles need calcium to be able to contract. Our sarcoplasmic reticulum have these openings called terminal cisternae, and these lie directly adjacent to T tubules. So we'll see on an image that we get a T tubule kind of spreading straight down vertically into our muscle fiber. And then on either side of that T tubule, we'll find a terminal cisternae of the SR. And so two terminal cisternae next to the one T tubule is what makes up our triad. And we'll see that again on our image. Myofibrils are bundles of our protein filaments. So this is what makes up our muscle fiber. And these contain the myofilaments that actually cause our contraction. So our myofilaments are arranged into our functional unit, which is the sarcomere. So we have many sarcomeres within a muscle fiber. Within the sarcomere, we're going to talk about two primary or contractile proteins, which are actin and myosin. Actin is referred to as the thin myofilament. Myosin is our thick myofilament. So remember those names, but also remember that thin and thick distinction. And so these myofilaments are part of the reason why our skeletal muscle shows up under the microscope as striated, because they stain slightly different colors. So we've said this already, our sarcomere is the basic functional unit. And so our muscle fiber is a muscle cell. The sarcomere is the basic functional unit of the muscle. If you remember back to our previous unit, we talked about functional units of compact bone and spongy bone. So for compact bone, our functional unit was the osteon, and it had many osteocytes within one functional unit. Our muscle system is kind of the opposite. Our functional unit is the sarcomere, and we have many sarcomeres that make up one muscle fiber. And so we have many functional units within each fiber. The Z disk is a network of proteins that act as our anchor point for our actin, our thin myofilaments, and this also acts as the border of our sarcomere. So a sarcomere goes from Z disk to Z disk, And we're really going to look at one sarcomere by itself when we go into this a little further. But really, our sarcomeres are lined up kind of one after the other from Z disk to Z disk. Okay, so a lot of these we have introduced already. A couple down here we have not talked about yet. We'll get into those a little bit later into the chapter. One thing I want to highlight again is this triad. So T for triad, T for T tubule, T for terminal cisterna. So two terminal cisterna, one T tubule makes up that triad. So our sarcolemma is going to cover our entire muscle fiber. And then we're going to have T tubules. This is really showing one, but we're going to have multiple throughout our fiber that are those inward folds of the sarcolemma. And that's how we can propagate the electrical signal that our nerve or our muscle is receiving from the nerve deeper into the muscle and our fiber. And then we have our terminal cisterna. Remember, these are the sarcoplasmic reticulum of our muscle fiber. And the main thing to remember about those is that they are releasing calcium. And like I said, we will talk about why that's important here shortly. One other thing that's good to point out is mitochondria are shown here as well. Remember, our muscles are always going to need energy, so we have a lot of mitochondria within our muscle fibers. Myoglobin is similar to the hemoglobin in our red blood cells that bonds to oxygen. So myoglobin is a way that our muscles can kind of store its own oxygen. And then we talked about this already. Our muscles have a really, really good blood supply, and they have a bunch of capillaries right on the surface to be supplied with even more oxygen. Okay, so we just talked about these a little bit, but this is what helps with the electrical component or propagation of that electrical signal, that nerve impulse through the muscle fiber. So it initiates at the sarcolemma, the transverse tubules, the T tubules help carry that inward. Those are the inward folds of the sarcolemma. And then our sarcoplasmic reticulum is the other part of the triad, the two terminal cisterna, and they're going to release calcium in response to that electrical signal traveling down the transverse tubules. All right, so this is just another good summary slide of our organization, just so we kind of all stay on the same page of what we're talking about. So remember, our tendon is what is responsible for attaching the muscle to the bone. We have an epimysium that is going to surround our entire muscle. Our entire muscle is made up of muscle fascicles, and these muscle fascicles are surrounded by a perimysium. Our muscle fascicles are bundles of muscle fibers. So our muscle fibers are surrounded by an endomysium. And so remember, that's just the connective tissue covering. That's just kind of like the saran wrap, the extra layer. The sarcolemma is the cell membrane. It has a much more important role in terms of cellular function. The endomysium is just the covering. Our muscle fibers are made up of myofibrils, which are made up of our actin and myosin myofilaments and all the sarcomeres. So when we talk about this sarcomere, we're really zoomed in into the myofibril. So here is an image that represents the structure of a sarcomere. And so we have a few different zones of our sarcomere that we're going to talk about. And there are, our next slide has some of the words that go along with this as well. So you don't just have to memorize everything I'm saying. Earlier, we said the Z discs are the borders of our sarcomere. So here's our Z disc. And they also act as the anchors for actin. So here's our thin myofilament actin. So those are anchored at the Z disc. Our I band here runs from the end of each myosin. If we kept going, we'd find another start of the myosin, the thick filament, over on this side. And so our I bands only contain actin and a Z disc. Our A band is where we get the overlap. So we have overlap of our actin and myosin. You'll see myosin has these kind of golf club head looking extensions that we call heads that we'll talk about that attach to the actin. And so that's the entire A band. And so you can see within the A band, we have an H zone where there's not necessarily overlapping going on while our sarcomere is relaxed. So you'll see how it looks when we have a contracted version shortly. And so our H zone just has this middle portion with just the myosin, and it also contains the M line or M zone, which is going to be our delicate filaments, and that's partly what helps anchor our myosin. So myosin, thick filament, actin, thin filament. So here's some of the words that go along with what we just talked about. So obviously this is an important thing to remember because we've said it a lot of times already, but the sarcomere is the basic functional unit of our muscle fiber. So this is the smallest part that contracts. And so what happens when our entire muscle contracts is that we have thousands and thousands of sarcomeres that are all contracting together to make that allow that muscle to function. So we said our I bands are going to stain lighter. That's just our actin and Z disc. Our A bands stain darker. So there's our striated appearance. And that's going to be the overlapping of actin and myosin, except for within that H zone. So remember, the H zone was the middle portion of the A band, and we had no overlapping. And then those chain links, the silver going down the center is our M line, which are delicate filaments, and that's the anchor for our myosin. So Z discs anchor actin, that's our thin. Delicate filaments anchor myosin, that's our thick. We do have some other filaments and proteins that help our make up our sarcomere. So our actin and myosin are the contractile proteins, but we do have other filaments contributing. So titin is one of those. And so this is a protein that goes with a couple of our characteristics we talked about in part one. So it makes muscles extensible. It allows them to stretch past their normal length and still function. And they're also help with elasticity, meaning it helps it return and maintain its resting length even after being stretched. And they actually look like springs, no surprise. Think of how a spring functions. So it's built just like the job it has to do. Structure always follows function. All right, if we zoom in on our thin filament, we'll see that we have active sites, and this is where our myosin heads, these golf club heads, are going to actually attach to the actin. And we're going to talk about troponin and tropomyosin. This is where our calcium comes into play as well, because calcium activates troponin to get tropomyosin out of the way of our active site. So there's a couple more steps. It's not quite as simple as myosin just attaching to actin. We have to open up the active sites for the myosin. In terms of our myosin, we call the longer part, the actual chain, is our rod. And then this golf club head is going to attach to our actin active site, and that's what's going to actually contract. So actin forms kind of a double helix structure, kind of two strands that are wrapped around each other, and they're going to be attached at either end of our sarcomere. Think in your head what those ends are called. So if you were thinking Z disc, you're thinking on the right track. So myosin and actin are going to bind with each other, and this can only happen at the active sites. But these active sites are not always available. So we have a couple steps to allow the active site to be open to myosin. So we have troponin and tropomyosin that are part of our actin strand. And so tropomyosin is the lid on the active site. It blocks the active site. And so it says blocks the active site on troponin. This should really say blocks the active site on actin. Troponin is what lifts the lid on the active site. So troponin is going to move tropomyosin out of the way. So we have calcium that comes in and binds to troponin. So calcium is attracted to troponin. When calcium binds with troponin, it causes it to lift the lid of tropomyosin, and now our active site is available. So this action, this release of calcium, the bonding with troponin, and the lifting of tropomyosin is how we regulate when our active site is available for myosin to bind to our actin. So without calcium, without our SR releasing that calcium, our myosin cannot bind to our actin because tropomyosin is blocking the active site. So troponin lifts the lid on tropomyosin. So calcium binds to troponin, troponin binds to tropomyosin and lifts it up. So our myosin is our thick myofilament. We said it has a long rod with the globular or golf club shaped heads. And when our myosin actually binds to the active site on actin, we refer to that as a cross bridge. We'll talk about the role of ATP in muscle contraction as well. So we have the presence of ATPase, which are enzymes that break down ATP. Remember, cellular energy is in the form of ATP, but how we actually release that energy to be utilized is breaking the bonds between our three phosphate groups of ATP. And so we'll see that in order for our muscles to reset and be ready to contract, we need ATP available. So this is part of the reason for the rigor mortis response. If any of you have heard that term, that's when muscles get really stiff after death. Because if our system shut down, if we're not making more ATP, then we are unable to release and reset our sarcomere for the next contraction, so the muscles just stay bound. All right, here's just another way to look at your sarcomere. So remember I said we zoomed all the way in to where we could really only see one sarcomere, but this is how they're going to be organized within our myofibrils. So they're going to be lined up from Z disc to Z disc to Z disc. And so here's another way to look at your different regions of your sarcomere. And we saw this not too long ago. Okay, the neuromuscular junction. So this is a neuromuscular junction under an electron microscope. So you can see our nerve is coming in and it branches off to various muscle fibers. So it's not necessarily that one nerve goes with each individual muscle fiber. Oftentimes our power muscles are going to have a neuron with a lot of different branches. So a few neurons to control a lot of the muscle. And then stuff like our fingers that have really delicate movements, our motor neurons tend to control fewer muscle fibers so that we can be more precise, if that makes sense. All right, I am going to go ahead and stop here for now since we got through our sarcomere parts and our new, a lot of new vocab terms. So circle back to that table that we looked at the very beginning of this lecture. Make sure that you can fill in all of those new words and understand our parts of our sarcomere, because that's going to be important before we get into our nerve and what actually happens once our muscle fiber and sarcomeres are contracting.